All right, guys, in this video, the first thing I want to talk about is the difference between R-selected species and K-selected species. These organisms we both often talk about in their role in succession, which is how the animals and plants in a certain environment change over time, how one group tends to displace another group. R-selected species usually tend to come in at the beginning of succession. Sometimes they're referred to as pioneer species. They come into a new niche, they reproduce very rapidly, sort of take over the environment and try to block out competition. Their strategy, again, is to reproduce fast, block out competition. They tend to have very short life cycles and give very little parental care. Some examples of good R-selected species would be like grasses, also insects. Typically, R-selected species are represented, at least their growth curves, their population growth, in what's called the J-shaped curve, as you can see here, or sometimes this is called exponential growth. They tend to have a very short lag phase when they're put into a new environment, and then they very quickly reproduce in what's called the log phase or exponential growth phase. Oftentimes, that is also followed by very rapid death. Again, those are called R-selected species, and they tend to come in at the beginning of succession. They're called pioneer species. K-selected species, on the other hand, when you hear K, I want you to think about carrying capacity. These organisms tend to have pretty stable population sizes at or near the carrying capacity. These organisms tend to come in toward the end of succession. Oftentimes, they're called the climax communities. These might be your hardwood trees, or maybe elephants, possibly even humans. They tend to have long lifespans. They tend to have a few offspring, but they tend to give lots of parental care and take very good care of them. They tend to be characterized by the type of growth curve we're about to see here, which is called logistical growth. Once again, we see a short lag phase where they're getting used to their new environment, an exponential growth or log phase, and then up the top to finish up the X, we see what's called the stable equilibrium phase. In this part of the graph right here, two pairs of things are equal or near equal. First of all, the birth and the death rates are close to the same thing here, and also the population size has reached or has nearly reached the carrying capacity. Oftentimes it will tend to fluctuate around that carrying capacity. So again, K-selected species are characterized by this logistical S-shaped growth curve. Another point we want to make about R and K-selected species are the type of what are called survivorship curves that they exhibit. So let me pull up a survivorship curve and show you how that works. In terms of survivorship curves, your K-selected species tend to be represented by what's called a type 1 survivorship curve. Most of these organisms live pretty long lifespans, at least in terms of percent of their lifetime, and then they all die somewhere near the maximum life expectancy. Again, we're talking about things like hardwood trees, elephants, humans. Whereas your R-selected species tend to have a survivorship curve that looks more like this. It's called the type 3 survivorship curve. Very early in life, a lot of those organisms die. Very few of them make it up to the 100% lifespan stage. Again, type 3, characteristic of R-selected species. Type 1, characteristic of our K-selected species. Another thing I'll briefly mention here is what happens if a population exceeds its carrying capacity. And that means there's too many organisms to be supported by either the space, the nutrient availability, the food availability, or that kind of thing. If the carrying capacity is exceeded, let's say this line that I'm drawing here is the carrying capacity for a particular species. Typically what happens if the population size goes over that, it will die back to and fluctuate around the carrying capacity, K. Now if the population shoots way high, way over the carrying capacity, and those organisms damage the environment, maybe kill some of the trees or kill some of the plants, that population may die down to a new lower carrying capacity. because now the environment can't support the same number of organisms that it originally could. Or the other outcome is that if they really wreak havoc on the environment, the population size may just completely go extinct. Again, those are three possible outcomes that may happen if a population exceeds its carrying capacity. All right, another thing I'll mention here is dissolved oxygen. The oxygen levels in a body of water are controlled by several factors. One of them is temperature. Typically, the way this works, higher temperatures mean there's going to be less dissolved oxygen in a body of water because gases tend to bubble out of very hot fluids. Another factor is movement. Typically, more movement means more dissolved oxygen in a body of water. Rivers tend to have more dissolved oxygen than lakes. Third factor, salinity, saltiness. Typically, more salt means less dissolved oxygen. And then a fourth factor is the amount of plant life in the body of water. Usually, an increase in plant life means an increase in dissolved oxygen because of photosynthesis. Producers, productivity means the production of sugars and oxygen. Plants do productivity, so they increase the oxygen levels. That's typically the case. Also, the time of day. Usually, bodies of water are going to have more oxygen during the times of the day when there's a lot of light. And because there's a lot of light, there's going to be a lot of photosynthesis, therefore a lot of oxygen dissolved in the body of water. Now, I want to briefly mention a problem called eutrophication. This happens when a body of water gets too many of a specific type of nutrient, usually nitrogen or phosphorus. In a normal aquatic body of water, those two nutrients limit the number of algae that can grow there because they need those two things to grow, and usually they're in pretty small supply. If nitrogen or phosphorus levels increase, usually because of farming or maybe septic leakage into the body of water, that limiting factor is not there anymore. And the algae grow out of control. We get what's called an algal bloom. Now, at first, that may sound healthy because there's a lot of photosynthesis going on. But most of it's going on at the top of the water, and that oxygen is going into the air instead of into the water. That mat of algae is blocking the light that would normally help the plants lower in the body of water do photosynthesis. But instead, those plants now die because they can't get light. Their bodies, along with the bodies of the constantly dying algae, get decayed in the water column. And decay is a process of cell respiration that actually uses up oxygen. So even though there's all these algae on top of the water, the oxygen levels are actually decreasing because the net productivity, the total amount of photosynthesis minus the total amount of cell respiration is actually less than zero down in the lake. And the body of water is constantly losing oxygen. So that the fish start to die, and the body of water becomes very unhealthy. That problem of nutrient enrichment, oftentimes brought about by farming or other human activities, is called eutrophication. And you definitely need to be familiar with that term. Another topic I'll briefly mention is symbiosis. And one of the most important things that we'll talk about with symbiosis is the endosymbiotic hypothesis, or sometimes it's called the endosymbiont theory. This is the idea that prokaryotes gave rise to eukaryotes. That small prokaryotes were taken in, phagocytized by larger ones, and instead of being digested intracellularly like would normally happen, the larger prokaryotes started using these things as maybe energy sources. And the two bacteria became so dependent on each other that they couldn't live without each other anymore, and that the smaller ones eventually evolved to become the organelles. And this is supposed to be the origin of eukaryotic cells. There's a lot of evidence for this. Things like chloroplasts and mitochondria have a double membrane, one of which, according to scientists, originated from the phagocytosis event, which brought them into the original cell. Mitochondria and chloroplasts have a single circular chromosome, much like the ones found in bacteria. And they also can do a process very similar to binary fission. They can reproduce themselves independently of the cell that they're living in. Some modern day types of symbiosis include mutualism. where it's a relationship when both individuals in that relationship benefit. When you hear the word mutualism, I want you to think about two specific examples. I want you to think about lichens, which are a fungus living with an algae. The fungus provides shelter, a place to live, and the algae in return, or cyanobacteria, can do photosynthesis and thus provide food for that lichen. I also want you to think about mycorrhiza. Mycorrhiza, the word literally means fungus on the roots. This is a relationship between a fungus and a plant, where the fungus helps the root be better able to absorb nutrients and water, and in return, that fungus is supplied with sugar. So it's a good deal for both organisms. Commensalism is a relationship when one partner in the relationship benefits and the other one isn't helped or harmed. This might be like the bacteria living on your skin. They get a free meal. You're not really affected. It might be like the remora, which is a little fish that tends to live with or sometimes attached to sharks. It doesn't help the shark in any way. It doesn't hurt it either, but it tends to get scraps after a shark is fed. The final type we'll mention is parasitism. And this is when one organism benefits and the other one is harmed rather. One benefits and the other is harmed. This could be like a tick and a human. It could be like a tapeworm and a human. It could be something like the plasmodium, which is the organism that causes the malaria. How that it gets into a human, it benefits in that it feeds on your red blood cells. You, on the other hand, are harmed and can be killed, depending on the situation. So those are the main types of symbiotic relationships. All right, another topic I want to mention is photosynthesis. You should know that there are three main types of organisms that can do photosynthesis. Obviously plants, but also algae and cyanobacteria. Oftentimes they're called producers or autotrophs. And sometimes we refer to what they do as production because they produce two main things. They produce carbohydrates, sugar, and oxygen gas. Photosynthesis happens in three main steps. The first step is called the light-dependent reactions. And in plants, that happens in a location called the thylakoid, which are a pancake-like disc inside the chloroplast. It actually happens on the membrane of the thylakoid. And during the light-dependent reactions, or non-cyclic photophosphorylation, three main products are created. Those are NADPH and ATP, both forms of stored chemical energy that originated from the sun. And in addition, oxygen gas, which is created by the photolysis of water. Water is broken down in this process to provide electrons to make it happen. So these are the three main products of the first stage of photosynthesis. Step two is sometimes called carbon fixation. And this is when carbon is grabbed onto by certain enzymes and prepared for the process of the Calvin cycle, which will use that carbon and the NADPH and ATP, the energy from those two things, to make sugar. Carbon fixation in most plants happens out in the middle of the leaf using an enzyme called rubisco. This enzyme is probably the most abundant enzyme on Earth. Its job, like we said, is to take carbon and prepare it for the Calvin cycle. And usually, this enzyme works very well. But it has problems when the amount of carbon dioxide in the leaf is too low. This usually only happens when a plant is having to close its stomates in order to conserve water, keep from desiccating. The main plants that have a problem with this are called C3 plants. These are most of the plants in Alabama. When they have to close their stomates, rubisco, instead of grabbing onto carbon dioxide like it's supposed to, will attach oxygen to certain organic products. That process is called photorespiration. And that's a big drain on the photosynthetic efficiency of C3 plants. Now, C4 plants, on the other hand, have an ingenious little solution to that problem. They have a different enzyme. And that enzyme at least the initial carbon-fixing enzyme in the C4 plants, is called PEP carboxylase. And what PEP will do is slightly different than Rubisco, and we'll show you how that works here momentarily. So PEP carboxylase is out in the mesophyll of the leaf. PEP will grab on or fix that carbon dioxide out in the mesophyll. It has no attraction for oxygen. So these C4 plants can close their stomates and conserve water without having to have the issues of photorespiration that C3 plants have. Once that carbon is fixed, though, it's transported to another part of the plant, a small enclosed area called the bundle sheath, where it's released. And inside this bundle sheath, we will once again run into Rubisco, which will prepare the carbon for the Calvin cycle. So Rubisco is found in all plants, C3, C4, and the last type of plant we'll talk about here in a minute called cam plants. But this C4 setup, C4 plants tend to be like grasses like Bermuda and also corn. They fix carbon in one place, the mesophyll, and then they transport it and do the Calvin cycle in another place, the bundle sheath. And this allows them to be more tolerant of dry, hot environments. Cam plants are the most adapted for those kinds of environments. Unlike most plants whose stomates are stimulated to be opened by either blue light or blue light typically, cam plants close their stomates in the light and open them at night. They fix carbon at night when it's nice and cool. They store it as an acid called malic acid, and then during the day they release it out into the mesophyll where they have Rubisco as well. And Rubisco will then go on and prepare the carbon for step number 3 of photosynthesis, which is the Calvin cycle. A good example to know of cam plants is the cactus. It's very well adapted for hot, dry environments. Calvin cycle, here's the basics of the Calvin cycle. It happens in what's called the stroma of the chloroplast. And very basic, it takes carbon through the process of carbon fixation, and it adds to that carbon energy, electrons, and hydrogens from products of the light-dependent reactions, namely NADPH and ATP. And when those things all get added together during this process of the Calvin cycle, we end up first with a 3-carbon sugar called G3P. Eventually, though, those G3P 3-carbon units are attached together to make either glucose or some other sugar, like sucrose or maybe even eventually starch. So again, photosynthesis happens in 3 main steps: light-dependent reactions of the thylakoid membrane, the Calvin cycle, usually in the stroma of the leaf, or the mesophyll of the leaf, let's put it that way, and then the Calvin cycle happens in the stroma of the chloroplast. And our last review topic of the day is cell respiration. Cell respiration's purpose is to oxidize glucose, or food, in order to make ATP, a nucleic acid nucleotide. ATP is the source of energy for pretty much all of our metabolic reactions. Enzymes called kinases will remove the 3rd phosphate from an ATP molecule and transfer it to other molecules. This will provide activation energy to help those things do their jobs. Aerobic cellular respiration happens in 3 steps. The first one is glycolysis. During glycolysis, glucose obviously gets split, cut. It also gets oxidized. And during this process, 3 main products are made. 3-carbon sugars called pyruvates, or pyruvic acids. Some electron carriers, NADH, which holds some of the energy that used to be in the glucose. And 2 molecules of ATP are netted during this process They're made in a process called substrate level phosphorylation. Most of the sugar's energy after the end of this stage is in the pyruvate. You should also know that glycolysis happens in every type of cell, and it takes place in the cytosol or cytoplasm. Step number 2 of this process is the Krebs cycle, or sometimes it's referred to as the citric acid cycle. This happens in the matrix of the mitochondria. And in this process, the pyruvate, or at least the products of pyruvate, are oxidized and the energy is taken out of these products. In between glycolysis and Krebs cycle, there is a small step called the pyruvate conversion. And during this step, the pyruvate is oxidized from a 3-carbon substance down to a 2-carbon substance called an acetyl group. This acetyl group will attach up with a vitamin or a coenzyme called coenzyme A, and that's actually the product that will enter into the Krebs cycle. And once again, the Krebs cycle's job is to oxidize this, which means its job is to get out energy, to get out electrons, to take away hydrogens. And during this process, some main products are made. The sugar at the end of this process basically is released as 6 molecules of carbon dioxide. We're talking about per glucose molecule here. There are 8 molecules of what are called NADH made between Krebs cycle and the pyruvate conversion. A couple of very similar molecules called FADH2s. And finally, 2 more ATPs through the same process that was used in glycolysis, substrate level phosphorylation. Most of the energy that used to be in the sugar is now in these NADH and FADH2 molecules from glycolysis and also from Krebs cycle. So the last stage of this process, sometimes it's called the electron transport chain. Sometimes it's referred to as oxidative phosphorylation. It'd be good to know both of those terms. Before we get into those, a reminder of some locations. Glycolysis happens in the cytosol of every cell. Bacteria, eukaryotic cells, plants, fungi, etc. Krebs cycle in a eukaryotic cell happens inside the mitochondria. It happens in the innermost compartment of the mitochondria, which is called the matrix. And then the electron transport chain for oxidative phosphorylation in a eukaryotic cell, it happens on this inner membrane. This membrane is called, these folds in this inner membrane at least are called cristae. In a bacterial cell where there are no mitochondria, this electron transport chain is embedded in the actual plasma membrane, the cell membrane of the cell. Which is very basically, here's how it works. The NADH contains most of the energy now that used to be in the sugar. Oxidative phosphorylation oxidizes NADH, which leaves behind NAD+, which will get used again for glycolysis and Krebs cycle. The electrons that are taken away from that and their energy are moved across a system of electron transport proteins called cytochromes. And as those electrons move, their energy is used to create a hydrogen ion gradient. H+ protons are pumped from the inner part of the mitochondria, from the matrix, to the space between the 2 membranes, the inner membrane space. And we start to build up a gradient of H+. These H+ want to get back to the matrix because that's the way the concentration gradient is leading. But they can't because they're positive, and this inner membrane is made out of phospholipids. But there is a way in, and that way in is our friend ATP synthase, which is a channel protein and also an enzyme. And when it opens, these H+ will rush in through that channel, and the enzyme part of the channel will phosphorylate ADP using energy from this moving gradient of H+. And that creates lots of ATP. On average, that creates about 32 ATP molecules per glucose. This process of creating ATP is called chemiosmosis. And the whole process that we see on the screen is oxidative phosphorylation. The process of oxidizing NADH and FADH2 and using the electrons and the the energy from this oxidation to create or to phosphorylate ADP in order to make ATP. Now that's aerobic respiration. And most organisms do aerobic respiration. There are a couple of really small things that get by doing only anaerobic respiration. These would be very small things that don't need a lot of energy, like bacteria, some bacteria, and maybe yeast. Humans do anaerobic respiration when we don't have enough oxygen to continue this process. One thing that we need to very quickly remind ourselves of, the role of oxygen in cell respiration is to take electrons away from this final cytochrome. Oxygen is the final electron acceptor. It will take away those electrons so that the process can continue. And in the process of combining with those electrons and some hydrogens, it will create water. But know that oxygen's role is to be the final electron acceptor. Back to fermentation, or what we might call anaerobic respiration. Aerobic respiration on average makes about 36 total ATP molecules per glucose. On the other hand, that's supposed to say ATP. Anaerobic respiration makes only about 2 net ATPs per glucose. So it's very inefficient when compared to our aerobic process. And the only place in your body where it's going to happen at a great rate is in muscles during extreme exercise. When your heart, your blood vessels, and your lungs simply cannot provide enough oxygen for your muscles to continue to make ATP. We only do this in a pinch, and when we do, we create a waste product called lactic acid. Other organisms, like yeast, for example, do a different type of fermentation called alcoholic fermentation, and they create 2 waste products. Those 2 are ethanol, drinking alcohol, and CO2. The purpose of both types of fermentation is to keep glycolysis going. We have a limited supply of NAD plus molecules. And if we don't oxidize NADH back to NAD plus, we'll run out, which means we can't create ATP, and we will very quickly die without ATP. So anaerobic respiration keeps our muscles going until we can get oxygen to them. Once again, I hope you've studied. I think you're all pretty well prepared for this exam. Good luck tomorrow. I'll hopefully see you in the morning before the test.